Thank you, and welcome you to the panel discussion, where we'll have Gustavo and Steve come up to answer questions about the challenge, along with Wendy Hugh, Alex Bisberg, David Noren, and Byron Long in my lab. All four of them competed and won the sub-challenge last year in Dream 8, and have worked on different aspects of the challenge this year. Byron's been working on integrating with the Synapse Sage's interactive collaborative website. Dave Noren's been doing the testing. Wendy and Alex have been working on the visualization platform for you. So with that, I'd like to open it up to questions from the audience. Yes.
Hi, Steve. Question about the RPPA data. So we know the cleaned-up data is available after supercurve and all that. Will the raw level data before supercurve, will that also be made available?
No. It would be, we can talk with you about it and we could provide that. It would be too massive amounts of data, I think, to give out the raw data for each slide, and people would need to have a proprietary program like Regine to be able to read the raw data for some of those files. So that would be a barrier, I think.
There's a question back here.
So, as a layperson who has no real background in this and is not affiliated with any academic lab, is there any place for someone like me in this challenge? Like, there are like groups that say, hey, I want an extra body, or is this more intended for like different academic groups at MD Anderson or something like that?
I'll speak quickly and then I'll give it to Gustavo to answer. I would say it's open to everybody, and the benefit is having people from diverse backgrounds compete. And additionally, forums like this, and there's also going to be later a hackathon, it'll allow you to interact with people. We'll send out a list of people who are attending, if everybody agrees, to have the emails open to people involved, to match up with teams.
So what is your background?
I have a background in artificial intelligence and informatics.
So it's an interesting question. Let me open the question a little bit more with respect to other trends in the area of challenges, which is the gamification of challenges. You take a problem which is well framed in a particular area, but you can translate it into something that could be operated as a game. Like, for example, we saw to understand the faults of RNA in a game called Eterna, or to try to help with protein folding in Foldit. So I think that some, in this particular challenge that we have, the AML challenge, we felt that there is so much domain expertise that could be leveraged that cannot be translated in that way, that is a little bit difficult to pose it in a way that is that you treat the numbers without knowing that they are genes or proteins and so on. So I guess there is a lot that will be lost. But I would be particularly interested in knowing whether, if you take the data as numbers that could be meaning anything without the need of knowing that they are genes or proteins or phenotypes or clinical covariates, I wonder whether you can win. Because in the old days of CAST, some of you may know critical assessment for protein structure prediction, which people were given the primary structure, the primary sequence of the protein, and they had to predict the three-dimensional structure. People who won were people who knew nothing about biochemistry in the beginning, and the physicists that supposedly know how things interact in space and have force fields and so on, they lost miserably in the first because they didn't have a good calibration of their force fields. So I think there is room for people like you to try, and it's an invitation to learn a little bit more about the disease and try to understand some of the domain information.
I think David.
Yeah, I just want to mention that I don't think it's been mentioned yet, but we have a wiki site, and we've done our best to make it very detailed, with just for people like you in mind, to really kind of level the playing field so that we can really bring expertise from all sorts of disciplines. to bear, their particular talents on this problem. And I think maybe we have the URL somewhere.
Yes, it's on the flyer that should be outside. Thanks, Dave.
Just two very simple questions. First, first, I didn't. Can you summarize the objective of the challenge again? My understanding was you were to find best prognostic factor which best predicting the outcome, right? That's the goal of the challenge. Second, how do you evaluate the results? Who's winning? What's the? I didn't get that from the presentation.
So I'll let Gustavo and Steve answer the first part, and then Byron has been working on the testing with Kelly at IBM.
So there are, I think, 3 clinical endpoints. Can you predict who's going to go into remission? Can you predict who's going to relapse? Can you predict how long people will survive?
Yes, and the remission duration is the second one.
So, but I think that the interesting thing that Steve has in mind when he's studying this data is also beyond the fact that you can predict the phenotype from the genotype distribution. Can you see what are the features that are interesting in order to determine, for example, potential targets for therapy? That is harder to score. We are not going to score. What we are going to score is whether people can predict correctly the phenotype. But hopefully, those people that can predict best the phenotype are also the ones who have found the features that are most meaningful. If you want to speak about scoring.
Sure. Initially, there will be released a training set of data for which you can create and calibrate your models. There will be weekly deadlines, and you'll be able to submit your new version of your results. And we will score those against a test set of data that we hold back. Each week, we will release the results and relative rankings of all the submissions, not only for the weekly period, but including all the previous periods also. So if you change your model, you'll be able to see if it was an improvement or not.
So those training based on some synthetic data, you have those training and initial, those are all real data, but something you have answered previously from previous challenges.
You're given a training set which includes all of the real data and the outcomes, so relapse, remission, duration, survival. And from that, you're asked to build a model, which is best predictor, given features and not knowing outcome, what is best predictor of outcome. And so then you're scored on your predictive of data that's withheld, and that data you're scored on whether you can predict outcome, the confidence that you can predict that outcome.
Okay, so on that note, I'd like to know, is the data sufficient, enough to evaluate, how good the algorithm is? Steve, you mentioned that initially you start with 511 patients, it's narrowed down to 323. So is that the total number of patients that available, and from the 323 you were split into the training and the validation, whatever.
So I'll give this to Dave Noren, who's been working rigorously since January on testing and figuring out whether we have a good training and test set.
Yeah, the 323, I think the number I'm working with is more on the lines of 200 or 300. So I think we did a little additional filtering than Steve had mentioned. But what we've done is we've taken 2/3 of the data set, and we've tried to ensure that the distribution of the 4-day clinical covariates and the 231 antibody readouts or proteins follow similar distributions between the training and the test sets. Not sure if that, I think you're asking me whether or not it's predictable.
If there's enough power.
So we did some preliminary testing here at Rice alongside with Gustavo at IBM and some of his colleagues there. And we do show that there is a level of predictability, but we won't, we don't want to say that what we've found is the best that can be done. We definitely think there's a lot of room for improvement.
Let me add something to this before going to Pavel. Is that there is another aspect to your question whether we have enough statistical power in the validation set to find the people who can predict without penalizing them because the distribution is broad, because there is not too many data points, and therefore the p-value might be only big if we ask a lot from people, to have to predict correlations very close to 1, even though that's not the case. I think that we don't have that problem. We have tested that you can, in many other challenges, that having 100 to 150 is enough to find people who have sufficiently good predictability, even if it is not really up there. There's a second point that I want to make with respect to your question, which has to do with the generic dream question that we ask ourselves. When do you know whether a problem is solvable? When do you know whether a problem is solvable or not? I believe that crowdsourcing, in a way, there is no proof of a negative, right? In order to prove something, you have to prove a positive. However, if you crowdsource a problem to 100 groups and none can get a statistical significant result, then you have a good indication that the data set with which you are trying to address the problem is probably insufficient. Now, if you have done it yourself, maybe 1, 2, 3 methods that you tried, it's hard to say. Maybe someone else has a better idea. But if nobody in the community, at least today, knows how to analyze this data to squeeze some information, then probably that means that we need to add something to the data set in order to answer that.
I have 2 questions. One, are we allowed to use external data in addition to what you provide? And second, can we use one of your supercomputers, IBM or Rice?
So yes, you can use any external, as long as it's not data or information, as long as it's not something from the data set. So if you've been exposed to the data set before, then you can't be a part of the challenge, but anything outside. And last year in Dream 8, that actually was the deciding factor between the winners of the overall challenge and the winners of the synthetic challenge. They used external data in public databases.
And yes, you could use IBM supercomputing power, Rice, but you have to pay for it. No, you have to ask for it, and we'll try to provide it if you lack sufficient compute power. There's one more thing I wanted to say with respect to what other data you can use. That comes with respect to the gamification issue. That's another thing in which using all the existing information about AML could be useful if you go back and try to see whether your prediction somehow is related to some prior knowledge. I think this is necessary because we are trying to solve not a game like some gamification people are trying to do in order to reach more people, but actually a real problem. And for that, anything is game, except for going to one of the surviving patients and taking a sample from him or her. Yes, yes.
So, and I guess I would like to really encourage people to use external data sources to better understand the proteins and how they interact with each other. From my perspective, the way that this will be most clinically useful is if we're able to tie back certain groups that predict, or that how they do based on their proteins, because those are targetable. Again, I can't make a 65-year-old 52 so that they will do better, but if I can identify what the key protein is in that older group of patients, then maybe I will have one of that long list of drugs that are in development, and I can specifically go back and target it to those patients. So I would highly encourage people to use external knowledge about what the proteins are, who they interact with in formulating their patterns.
Yeah, I just wanted to speak to the visualization. It's actually another tool that can help you sort of understand what's going on with connections in the data. So as you saw, you can segment the heat map and just see how patients are actually surviving. So that allows you to check the test data set and see, maybe give you some clues or some ideas into where you might want to start.
Hi. While the visualizations are really cool, my question is mainly on the data sets. How do you put together your data sets for these dream challenges? Are they usually from a single institution, or are they collected from a number of different institutions?
This one is from a single institution.
What about for the other ones?
How do we put together the data sets?
Yes.
For example, the Alzheimer's disease challenge, we have 3 sources. One is ADNI, Alzheimer's Disease Neuroimaging Resource. It is public for everybody. So we cannot use that as a test set, because that's public. You can go and get it. So that's our training set. But then we have 2 other groups that have contributed to specific test sets, and we had to coordinate that the covariates are the same so that we can predict what and those are groups in Europe. There is another challenge that we have been doing, rheumatoid arthritis challenge, trying to predict the outcome of treatment on patients. And for that, we also use a lot of data sets, which implies a lot of data governance, a lot of MOUs, a lot of ways in which we make sure that we are not infringing any privacy issues, IRBs and so on. It can be done. It's a pain in the neck, and we have done it for several challenges, yes. But what is interesting also in the RA challenge, just one more thing, is that there was one consortium that said we want to produce a data set, basically measuring the SNPs, that could be used as test set, and that's what we are using as test set. So people can take some interest and say, I want to fund or I want to do the work for an extra validation set that will be used, and that has happened several times.
So, going back to if you're getting data from multiple places, and in some cases this actually even within a single institution, how do you normalize the data? Because people collect it with varying, definitions. Different machines have different calibrations and things like that. How do you normalize this, and thereby not getting into the same issue that I think you mentioned on one of your slides, which is, are we missing some of the potential drugs because we are discarding these drugs as not being useful in a certain population set. If your data is kind of getting mixed up because of difficulties in normalization, it's not an easy thing. How do you overcome those kind of challenges?
Right. So let me take a stab at that, and maybe the rest of the panel can also contribute. Sometimes I hear that organizing a challenge seems to be easy, right? You get data, you make a data set, a website, and that's it, and then you score. It's much more than that. As you see, there was a huge effort in visualization in this challenge. There was a huge effort in dry runs to see whether we had signal. There were efforts in different challenges on batch correction, whether there is batch effects, whether for example, in the rheumatoid arthritis challenge, there are 2 different scoring ways of determining how well a patient is responding. One is the EULAR, one is the DAS28. So we needed to see whether there is correlation between those, because we want to use one for one purpose. What I'm trying to say is that we look at this and we agonize about this. And I have decided not to do challenges because the data was so noisy or so hard to do the batch corrections or the different artifacts that could come with the data. And we value, I see many of you hopefully will participate in this challenge. I feel responsible for your time, because if you are working on it, even though you are learning something out of it, but you are given garbage data, then it's my fault, because you don't have to know it. You take the data as you get. But we do agonize about it, and we try to make sure that there is very stringent quality controls on the data and batch effects. Anybody wants to add?
This particular data set has been rigorously tested, both statistically tested at MD Anderson and then tested and trained with both BioLab and IBM. And one more question, and then we'll break for lunch so everybody can have lunch.
Sorry to get everyone from lunch. So be very practical. Is that only the winners are allowed to publish papers with the data set? Or is that if we're not the top of the top selected, we can still publish our algorithms models with the data set by citing the competition and the institution?
So short answer is yes. We can publish on the data set.
Oh, yes.
On the algorithm.
Right, so yes, we want that to happen. We want people to use the data. But for that, first you have to ask Steven, because he's the owner of the data, whether he is satisfied with his own publication. After that, the data will be open for everybody to publish. We also want to publish the result of the paper, of the challenge, for which we ask for people to wait until they publish their methods, especially if they are going to use the result of the challenge as saying, you know, we won or we got second. So we would like for them to wait a little bit until they publish that. Now, if it is just their method, without the data, of course, right? But, you know, David told me something that has to be said. There is, we will have had another guest here from Sage BioNetworks, but Stephen couldn't come and Thea couldn't come. So all this work we did also with a very important institution, a sister institution. Actually, we have partnered. I say that we married Sage BioNetworks, Dream married Sage BioNetworks. And so in a way, what we are trying to do is to use their strength, especially their platform called Synapse, in order to run and score the challenges and make people collaborate through their collaborative platform. And so this wouldn't have been able also without the Sage BioNetwork folks. One more thing that I don't think that we mentioned. We had another partner, which is Kornblau, right? Why don't you mention that?
Yes, PLOS Computational Bio has already agreed to partner with this Dream Challenge so that we know we will have at least one paper that's collectively published in PLOS Computational Biology from the winners and people involved. So that's a really good, very positive response. And that was very quick. They got back to us within less than a day. And any other publishing partners, any other incentives will be listed on the website. So with that, I'd like to thank everybody up here, Gustavo, Steve, Dave, and welcome you to the lunch which is outside. Thank you all.
We would also like to take this opportunity to invite you to the Dream 9 Hackathon. This event will be hosted at the BioScience Research Collaborative Building at Rice University in Houston, Texas, and will be broadcasted over the internet. The hackathon is designed for participants to share their ideas and approaches in order to receive feedback from other contestants, form collaborations, and possibly join efforts in the Dream 9 Challenge. All hackathon participants will get a chance to present on their ideas on-site or over the internet during the Saturday morning session. The afternoon session will be reserved for networking and presentations on different software that could be useful in this challenge. During the Sunday morning session, participants will get a chance to again present on new developments and input received, while the afternoon session will be again reserved for other constructive presentations and networking. An abstract book will also be provided for all the participants. So please join us for the Dream 9 Challenge Hackathon on July 26 through 27 for valuable input and networking. Thank you.